the MfPH Next Generation Seminar Series and Networking event for early career scholars, junior modelers, and epidemiologists working on or interested in mathematical modeling of infectious diseases and other threats to public health. Bienvenue à tous, la série de séminaires MfPH Next Generation est un événement pour les chercheurs en début de carrière, les modélisateurs juniors, les épidémiologistes travaillant ou intéressés par la modélisation mathématique des maladies infectieuses et d'autres menaces pour la santé publique. First of all, we wish to acknowledge this land on which the Fields Institute operates for thousands of years. It has been the traditional land of Huron-Wendat, the Seneca, the Mississaugas of the credit. Today, this meeting place is still the home to many indigenous people from across Turtle Island, and we are grateful to have the opportunity to work on this land. Premièrement, nous souhaitons reconnaître cette terre sur laquelle l'Institut Fields opère depuis des milliers d'années. C'est la terre traditionnelle des Huron-Wendat, des Seneca, des Mississaugas. Aujourd'hui, ce lieu de rencontre abrite encore de nombreux peuples autochtones de toute Turtle Island et nous sommes reconnaissants d'avoir l'opportunité de travailler sur cette terre. Our guest today, Professor James Watmough, is an associate professor from the University of New Brunswick at the Department of Mathematics and Statistics. Professor Watmough earned his PhD in mathematics from the University of British Columbia, and ever since has embarked on a journey of relentless exploration and discovery in the field of mathematical biology. Throughout his career, Professor has published groundbreaking research that reshaped our thinking in mathematical biology with more than 13,000 citations. His publications have been instrumental in inspiring a new generation of mathematicians and biologists. Notre invité aujourd'hui, professeur James Watmough, professeur agrégé de l'Université de Nouveau-Brunswick, au département de mathématiques et des statistiques. Professeur Watmough a obtenu son doctorat en mathématiques à l'Université de la Colombie-Britannique et depuis s'est lancé dans un voyage d'exploration et de découverte incessante dans le domaine de la biologie mathématique tout au long de sa carrière. Professeur a publié des recherches révolutionnaires qui ont remodelé notre réflexion en épidémiologie mathématique avec plus de 13 000 citations. Ces publications ont contribué à l'inspiration d'une nouvelle génération de mathématiciens et biologistes. His talk will be on reproduction numbers and final size relations for disease transmission models. Sa présentation portera sur nombre de reproductions de relations de taille finale pour les modèles de transmission de maladies. I hope that my introduction was not so long, but Professor deserves more and more long introduction. Please join me in a warm welcome to Professor James Watmough, who will share his invaluable insights with us today. The floor is yours, Professor.
Ooh, thank you. Thank you for that introduction, Idris. Yeah, you have to give an even longer introduction to Julian next week then. I think his talk is next week, right?
Yeah, sure.
Yeah.
It will be in 2 weeks.
Oh, in 2 weeks. Oh, okay.
Yeah.
Yeah, so I was hoping to give a little bit of an extension of overview of reproduction numbers and things for disease transmission models and other models. This term turned out to be far busier than I expected, so I didn't make as much progress on getting this talk ready as I thought. So this will have, so I've given a talk like this, I think it's been 2 years now. It was in 2021 for the Fields Institute. So a lot of the slides are similar, but there'll be a bit of variation because I had too many slides then. So we'll go through it in a different order. But some of you may have seen some of these before. So I'd like to thank, before I get started too much, thank the places I've gotten funding with. And this is of course, the MfPH, but also Omni Réunis, so the EIDM networks. And through various collaborations with people, CIHR and NSERC, and New Brunswick Health Foundation. And I always like this, I know this is the MfPH seminar, but I always like the logo that Omni Réunis got for their network. I think it was unintended, but I really like the way it's so that there's a One Health logo. or a logo for modeling in One Health. Now, I need to find my pointer, sorry. That's my pointer, right? Yes, sorry, I was pointing with my mouse, which you can't see. So this little dot here is supposed to represent people. This is supposed to represent animals, animal health. And this is plant health, environmental health. And so this was a little stylistic logo that they came up with to put these together. So this is I think this is relevant for thinking about the reproduction number more generally, because it is something that appears in ecology, epidemiology, and virology, and in plant ecology and animal ecology, and all of these diseases that we've been looking at, all of these pathogens we've been looking at, they have components in all three of these, right? And so we're tying these things together. What I really like is this little stylistic, what is it, an M they have? Looks a lot like the stylistic S, I suppose, around the CCDM logo, the Center for Disease Modeling, which is, the Center for Disease Modeling was established after the first SARS pandemic in 2003, with help from Zhenhong Wu and Pauline, whose name I saw here, Pauline van der Driesch, and Fred Brouwer. And so that's still going on at York, and we have a Canadian network of that. So that's where I've been getting a lot of my collaborations with. But this little serpent is supposed to be, of course, the worm for schistosomiasis, I think, right? So it's the pathogen. And I kind of like this logo of Omni that brings in this pathogen that connects everything together. And so that's a bit of the theme I have for this talk, is that we're interested in diseases in people, right? Most of us, but we have to consider the environment and animal health and animal diseases, because all of these pathogens bounce back and forth, and we're really following the pathogen, right? And I have to remember, sorry, I have not given a talk on this computer setup for a while, so I might do some strange things. So looking at the reproduction number, it actually goes back quite a ways, but one of the first people to notice its importance, I guess, was Lotka back in 1939, 1930s. And there's a nice little summary of all that in a paper by Dietz. And it comes out of essentially demography, I guess. And so the history of this number essentially is demography and ecology, but for some reason it fell out of favor in ecology in favor of eigenvalues. But it ended up getting used a lot in disease transmission models. And so it's come to be something that we use in disease transmission models, but it's really an ecological creature, right? And so in the demography side of it, it's nice and easy to think about the reproduction number. M is think of it, the rate of individuals giving birth at age A, and then you multiply that by the probability that they survive to age A. And then if you integrate over all the age groups, you get the total births per person over their life, right? Which is what the reproduction number is. So this gets called, I think, the net maternity function. And then you get variations on this in demography with the reproduction number. And the one we end up seeing lately in the news is I forgot to look to see what it was called. They always talk about this number 2.1, which is the replacement ratio, I think, or something like that. That as a human population, if families don't have
the replacement rate in terms of fertility.
Is that the replacement rate? Okay, yeah. I couldn't remember which ones. Yeah. So that's a little bit different because that's not following through survival up to a certain age, right? So it's a slightly different setup, which is why it is the 2.1 rather than just the threshold of 1, right? And also the half of the births are male, half are female type thing, right? So there's this all kinds of connected numbers that you see, but essentially somewhat together. Thanks, Michael. Right, and so all we do when we do epidemic modeling is replace that with, or in the context of epidemics, is replace the maternity function with a rate that infected individuals produce new infected individuals, right? And I always think of this, I don't know why I think of this best in terms of an integral, integrating over ages or integrating over some aspect or different states, right? Right, and so we need to, if we want to use this reproduction number in any sense, we need to set up essentially birth of new infected individuals over states and the probability of survival. Yes, I was trying to think where my next, oh, there we go. So we interpret this as an expected number of secondary infections arising from a typical index case. That's the usual definition given. And so always keep in mind that this is an expectation, right? Some kind of an average. And this notion of a typical individual is always problematic, right? And so I'm going to try and touch on different aspects of that as we get going. And then in the abstract setting, we can just set up a evolution equation of some sort or some kind of matrix equation and talk about a next generation operator, and it turns out to be an eigenvalue, right? So we'll end up looking at that. Now, one thing that I still haven't quite wrapped my head around, and okay, some of these slides are, I'm getting a preview of the next slide, which turns out not to actually be the next slide. Okay. Sorry, I haven't used this platform for a bit and I'm just a little confused. I think I got it sorted out now. Idris Azara, I see a panel sitting on the top of my screen. Is that blocking your view too?
No.
No, okay, that must just be, it's my little annotation controls for Zoom, so you don't see it, that's good. Never mind then. So yeah, I also hear about this effective reproduction number, which it's been around for quite a while. It was introduced, I think, by, well, at least in the SIAM, that classic SIAM review paper of Herb Heathcote's back in 2000 was mentioning an effective reproduction number. And we saw this a lot, we still see this a lot in the literature and in the news and in reporting of what's going on. And it always baffled me a little bit. And we also see this ecology as effectively an eigenvalue of a projection matrix. So I think there's a connection there. But it's defined as the actual average number of cases generated by one case in the presence or absence of partial immunity, various odds and ends, things like that. So this is taken off the science table dashboard a while ago. So mathematically, this is an eigenvalue resulting from a linearization. But the reproduction number, the basic reproduction number, the one that we get from our mathematical models is usually linearized around an equilibrium, right? And I've always been a little bit baffled about what to do if we're linearizing our solutions, our data around something that is not an equilibrium. But we do it anyway, right? So I'm just, I'll say what this, say a bit about this effective reproduction number, but the long story short, I don't really know what to do with it or how to interpret it. So we calculate it, right? And say whether it's above 1 or below 1. But since we are not linearizing around an equilibrium, in the mathematical abstraction sense. This effective reproduction number, it tells us a bit about where we're headed. It's kind of like a slope, right? But the fact that it's bigger than 1 says we tend to be growing now, but doesn't say anything about where we're headed, right? And so we do see these popping up and down below 1. But I've never really been sure what to make of that. So I won't focus too much on the effective reproduction number other than to mention it, so we can put it into some perspective with the reproduction number. And maybe I will eventually get educated on why it gets used so much. The other aspect, this notion of well, we need to count up somehow the reproductive capacity in terms of new disease burden or new infections, new diseased individuals or infected individuals, depending on how we look at it. And at least for a lot of the respiratory infections, or at least the respiratory infection we've been fighting with for a while, and the ones that are running around now that seem to be wreaking havoc in my classroom, which I think is RSV and probably influenza, various leftover strains of coronaviruses, and then COVID. I don't know, students seem to be getting permanently infected with some respiratory infection this term, always circulating around. In any case, if you just look at the viral load of an individual, you can make a connection between symptoms and virus shedding. And at least for influenza and coronavirus, you get that the viral load peaks and then the symptoms kind of lag that, depending on all kinds of factors about how much your immune system kicked in, whether you've been vaccinated or not, comorbidities, all kinds of things, right? But roughly, you can use viral load as a indication of infectivity, and there's some connection to disease, right? So the whether you end up as a case of a disease or not, right? But of course, this is going to vary a lot between individuals, right? Especially when you start to have people who are vaccinated, unvaccinated. The vaccine may have had a different effect. People who now have hybrid immunity, we've always had hybrid immunity to things like influenza, well, at least if we've been getting vaccinated. But you have all kinds of variability in what the viral load in response to infection looks like between different individuals, and the connection between the viral load and symptoms and disease, right? So there's this heterogeneity between individuals that comes in when we try to talk about a reproduction number in a typical individual, right? So I've got to give my plug for heterogeneous models and individual-based models there. And the other side of it is probably the environment, but also a bit the individual. So this is back from SARS, the 2003 strain of SARS that also kicked off a lot of epidemic modeling in Canada. And set up a lot of the basic models that got grabbed for the 2020-2019 outbreak, right? Or pandemic. We see here the huge variability between the number of secondary infections people cause. So this, I like this just because the extremeness of it, and it's a picture I happen to have handy. So this individual labeled 1 has infected all of these individuals here, well, the ones directly connected by the arrow, as the secondary infections caused by person 1, and then they've also infected person 6. So person 1 has infected a lot of people, but most of these people don't pass on the infection to anyone else, or maybe just to one other person, right? So this individual caused a lot of infections, but the tertiary infections didn't explode in the same way, right? Except for this one here, right? Which produced, so number 6 produced a lot of tertiary infections, I guess, 1 being the primary, 6 being secondary, and then the ones that 6 infects being tertiary. But a lot of those just produced a few, right? So you get these big bursts, right? And I think in the case of SARS 2003, it was a mix of how the individual actually responded and what was going on. There's a lot of within hospital transmission. So sometimes a lot of close contact. So it also depends on the setting, right? In certain conditions, certain locations, the pathogen transmitted really well and the person was producing a lot of pathogen that survived, right? I think we saw this with the, when was it? Late 2000 or early 2001, there was a meatpacking plant in Calgary that had something like 2,000 cases. So pretty much half the cases in Alberta were caused by one location in Calgary, likely because of I haven't looked back recently, but I think at the time the conclusion was the temperature was right. The group of people that the transmission was happening among were co-workers who were also living in the same vicinity, sharing transport, right? A lot of carpooling. And so it just, the pathogen had the ideal environment and population to transmit in, right? So my point there is that there is a lot of variability between individuals and between settings where the infections happen, right? And so this gets back to what are we going to, how do we incorporate this into the reproduction number? What does it all do? Things to think about. Now, so I'm going to show you some contact tracing networks from, can I call them networks? Yeah, contact tracing network. Well, the pictures of some of the results of the contact tracing done in New Brunswick. So keep in mind, New Brunswick has a population of roughly 800,000 people, right? Spread almost uniformly all over New Brunswick. Our largest city is Moncton, which is now bigger, but I think when I looked at this a year and a bit ago, the population was still around 80,000. And Fredericton, the next largest city, I think, was 60,000. And we have St. John, which is also comparable, right? So 3 cities that are about the same size there, and a 4th city up in Miramichi that's not much smaller, right? So very, very different than Ontario, right? Or Manitoba, where you have bulk of the population in one place, right? Spread around quite a bit. But we got these really neat networks. So zone 3 was a certain region. I believe that might be the region around Fredericton. And these are just a few of the cases, few of the samples of the contact networks that came out in the middle of 2000 or maybe early 2001, I think, I can't remember. And I kind of think some of these are neat. So these are essentially, people are doing the contact tracing. They have people who have symptoms, right? So not just people, or not necessarily all the people who are infected, right? Not even necessarily all the people with symptoms, but the people they picked out as having symptoms and having a definite infection. And then they do the contact tracing and the interviewing and try to connect cases together, which of course isn't directional. All they can do is say that these 2 cases are connected. They can't always say with any certainty who infected whom, right? And so you end up with these clusters, right? Households. Everybody was infected. We're not sure who infected whom. And then households connected to other households, right? But the one I really like, that didn't work. Why did that not work? Oh, it did. I put a graph up. Yeah, I don't think this is too relevant. That's just a distribution of the degree and the cluster size. This is the one I really like. This is the result of contact tracing, and I never did quite figure out what the setting was, but essentially they were certain that these infections were all connected essentially in pairs here, right? But it looped in on itself. So they couldn't really decide where it started, where it ended, or turn it into a line, which I thought was kind of cute. The main message here, though, is the different patterns of cluster sizes, right? And different degrees the distribution of the degrees of the infection, right? Think about just to sort of remind us everything that goes on about the variability between individual infections, right? And between individuals. Okay. Right. Yeah, and I didn't say this at the beginning, but I will keep talking, but feel free to just interrupt, which means, and you might want to just shout because I don't think I have, wait. Aha, sorry. Yes, sorry, I just realized how to get my chat up. Yes, and Martin, yeah. So yes, so SARS-1 was nosocomial. A lot of the transmission was in the hospital setting. And so that specific aspect of the setting, I believe probably also in combination with the individuals who were there, but a lot of the way that the individuals were laid out in the hospital wards, how they were being treated, and the intubations, yes, led to lots of super spreading events. And there was one that was really interesting in a hotel, which I think was a connection of several rooms by outdoor plumbing. Yeah, but so the idea there is that the setting matters. Certain settings led to really rapid transmission, which led to super spreading events or super spreading individuals, right? Which means there's a lot of variability between individuals and types of ways the infection can happen, right? Yeah. Oops, and what am I doing here? Hitting random buttons. So just quickly, I'll probably scoot through this a little bit quick to get to the other interesting things I wanted to say. So if you go back and look at the original papers of Kermack and McKendrick, there was a integral equation model proposed, which is really kind of nice to look at and gives you a nice idea of how the reproduction number and the final size come out of that. And it lets us connect the, so here, X is the incidence of new infections. And so that's the number of people that were infected a certain time ago and are now A days, say, or time A since their infection, right? So it's like a cohort of people infected a certain number of days ago. These people will have a certain probability P of surviving to that age, right? And still being infectious. And their infectivity, their reproductive capacity in terms of the spreading of the virus or disease to new people would be measured by this beta, right? So if you integrate over that over all past ages, and then this infectivity is per effectively multiplied by the number of susceptibles, really multiplied by some function of the number of susceptibles and size of the population, right? Depending on, but if you just assume a sort of a bilinear incidence, if you double the number of susceptible individuals, that bilinear incidence assumes the number of cases will just double, right? Grows in proportion to the number of people who can be infected. And you get this nice little equation here. And I think I will, there we go. Yeah, and so the I that you used to in the simple SIR model, the differential equation model would just be the integral over all ages of the incidence a time A ago times the likelihood that you can survive or still remain infectious at a time at an age A, right? So infection age. Yeah, so that's all in that one paper of Kermack and McKendrick, and quite a nice little paper if you haven't looked at it yet. And it was reprinted in, I've forgotten, either a SIAM journal or the Bulletin of Math Biology, forgotten which one. And I think I will just skip ahead a little bit. Yeah, in the interest of getting up. And so you end up with a reproduction number in that perspective that is integrating over all the ages the likelihood of survival to that age and the rate that people of that age are producing infection, right? And so that's the basic idea coming out of that. And then in the ODE part, which I think I will focus on in the interest of getting something focused on how we think about this reproduction number and the heterogeneity and different things that can happen in terms of a simple context here. So if we divide our population into N, or infected individuals into N stages, or even just N types, right? N types of infection. And then we have a vector of populations in each stage, and we can set up the differential equation or the subset of your differential equations that look at how the numbers in those stages change, right? And so the changes are going to be because of infections, right? I'm pointing with my mouse instead of my little laser thing again. It's going to be with the number of infections, which we can set aside as one piece of it, and then progression through various stages of infection, right? Or age, or whatever else you want to put in your model, right? And so I'll call this one part here a transition matrix and the other part an infection matrix. And different explanations of this use different signs on the matrix. And I don't think I wrote down this reference, but there is a, Jim Cushing has written some really nice overview papers on this from a discrete time approach in the context of ecology and demographics. It's really nice. Right, and so for the simple SIR model, you have two states of infection. You can be exposed or infectious, and you just subdivide everybody into those two states. And then you can build these two matrices essentially from your thinking about how people progress through the stages and how infectious they are at each stage. So we're saying here that in this matrix, the first column and first row correspond to the exposed or latently infected individuals. The second column, second row correspond to infectious individuals. And so this beta S0 here, S0, is because the infectious individuals produce offspring who are in the exposed state, right? And nobody is born in this SEIR model. No one is born, or nobody becomes infectious immediately upon infection, right? You start with the E state and move up. And then the V is just how do you progress through, right? You, the off diagonals here, you move out of the E compartment into the I compartment at some rate, and you can't get this 0, you can't get back from the I compartment to the E compartment, right? And the diagonals, I'll mention this again in a second because it's a really useful little piece of information that doesn't always get connected in when you learn this. We have two ways out of the E compartment. Either you progress from latent infection to fully infectious, or you leave the population, right, by whatever means. And so this K plus U and the alpha plus U are adding up the ways you leave compartments, and the off diagonals are the rates that you transition between, right? I'm not going to dwell too much on that. I'm just going to say that, so we'll make up some of these matrices and think about more how these matrices, how we can construct them, but we won't go into too many details of what's going on in the ODE model behind it or things like that. So then we can take the inverse and the analog to the integral in that integral equation is that you end up with looking at the eigenvalues of this matrix, fecundity times the inverse of the transition probabilities, transition rates. And so this one has a 0 eigenvalue and a funny looking eigenvalue here that we're all familiar with as the reproduction number, right? Huh. Wants me to jump to latest, but I'm on the latest. Okay, and I'm just going to say this really quickly here, just in terms of parameterizing and thinking about the parameters in your model. The ODE models are all underneath there is essentially an assumption that the time spent in each stage is exponentially distributed. And so when you have this rate, some rate that individuals leave this stage, either to transition, oops, sorry, I'm using the wrong pointer again, either to transition from exposed to infectious or latently infected to infectious, which is happening at a rate K, or to leave the population at this rate mu. The underlying assumption of the differential equation ends up being that the time spent in stage E before leaving is exponentially distributed with a parameter that's the sum of the rates that you leave, so mu plus kappa in this case, right? And so you can use 1 over mu plus kappa as the expected time spent in the compartment. And the other handy little piece, if you're not aware of it, is if you take the ratio of one of the exit rates divided by the sum, then you get the fraction of individuals that progress to infection, right? I'm guessing that's not new for very many of you. Just looking at my time, thinking about where I want to get to. Yeah, okay, I think I might skip through that because I was going to focus on the next generation matrix, but I guess, yeah, I guess we will. No, I can skip that, yeah, and get to skip some of these technical details and get to some other pictures here. So yeah, so having said that, and this is not new for almost everyone, I suspect, right? So the interpretation of this is that the kappa over kappa plus mu is the fraction of individuals who progressed from latently infected to fully infectious before leaving the population, right? The beta times the S is the rate that these infectious people produce new latent infections, right? Proportional to the size of the susceptible population. And the 1 over alpha plus mu, that reciprocal of the rates that we leave the infectious compartment, is the expected time people spend infectious, right? So this makes a nice little measure of reproduction. And it turns out to be the eigenvalue of this matrix FV inverse, which I skipped the details, but is connected to the stability of the differential equation, right? And I think this is something I wanted to get to. And because I have too many term tests to get ready this term and too many people coming to office hours, I didn't quite get this completely filled out. But let's just think about how we can break this down and think about how to separate this out into infections and so new offspring and transition between states and what these states should be. So this is a classic in-host model. The T is the target cells. So these are uninfected cells, host cells. The E are the latently infected cells, right? And so they've the virus is, you think of this as being your lung tissue, right? T is the cells in the tissue that are susceptible to the virus. V is going to be the virus that's in that tissue or near that tissue, but not in a cell, right? So intercellular virus, free virus. And the E will be the exposed. So you get this encounter between target cells and virus, and your target cells become infected at that rate. So we have this transition from an uninfected target cell to an infected target cell at this rate beta. There was supposed to be a subscript 1 on there, but that doesn't matter. I think a subscript 1 will appear later. That should be here. And then we have, similar to the SEIR model, right? We have a progression at rate K from latently infected to infectious, right? So inside the host, if you have the cell that is infected, the virus works its way inside the cell, and then it just takes a bit of time for the machinery to get going for the virus to start replicating itself inside the cell and for new virus to appear on the surface of the cell and either enter intercellular space or by lysing the cell or by budding off the surface. So the TEIV, the E is always, I never did like the E, but I still find I write it down. So T is target cell. Think of the susceptible cells. E is, it should be a latently infected cell. It's, I guess E, so E in the SEIR model doesn't make sense because exposed and latently infected have different meanings in epidemiology. But the E here does make some sense because this is actually the eclipse phase of the cell. But think of it as the latently infected cell. The virus is in there, but it's not budding off the surface. It's not an infectious cell yet. The I is an actively infectious cell, and the V is the virus. And sorry, I'm using V in 2 different ways, but that's okay. You'll see what I'm doing here. And so this term is a switch from, the beta TV is a switch from uninfected to infected cell due to an encounter with the virus. So the virus binds to the surface of the cell and infects it, right? The A is a replenishment of the target cells, which typically ends up in these types of models, in the in-host models, as either being some logistic or just a constant. And so this, you can think of the A minus mu1 T as just the background appearance and disappearance of susceptible cells for reasons unrelated to the virus, right? The mu2 is the rate that the, so the mu1, the mu2, the mu3 are all rates that the cells are naturally removed. I think in most cases pretty small compared to what the virus is doing. And this kappa is the progression from latent infection to infectious, which in this context means the onset of the cell producing new virus, right? So this is a production, this is a switch to productively infectious virus. And this P, so I really want you to think about the beta and the P here. This P is the production of virus by the infected cell, right? And then here, the virus is naturally cleared, right? It doesn't stick around forever in your, if it's not inside the cell, it gets naturally cleared. Ignoring all the immune responses in here, right? And this beta 2, since infection happens effectively because of virus, free virus binds to an infected cell, or sorry, binds to a cell, enters it, the virus disappears from this pool of free virus and becomes a virus inside a cell, right? So effectively, the virus encounters a cell, and the E is a virus plus a cell, right? Or maybe several viruses, depending on how many did the infection. 47. Okay, I should wrap up here soon, shouldn't I? Right, and so, but I've labeled this as a beta 2 rather than the same beta because you don't always have to measure virus the same way you measure cells, right? So the units might be different. And there are other things that can go on that you might have multiple viruses binding to a single cell. But in any case, this is somehow part of the infectious infection process, right? So the reason I wanted to sort of settle on this model and probably just in a couple of minutes just wrap up with this, thinking about this model, it's kind of nice because I've been getting people have been asking me and essentially complaining that in a lot of these models, the reproduction number is ambiguous. If you go back to the way that you construct this, you effectively need to look at this model and interpret what it means in terms of which things are new infections and which things are transitions, right? And there's flexibility in how you do that, which can lead to different ways to write down these reproduction numbers. So the piece here, I think, is, and if I was clever, I could do a poll, but I have not figured out how to do that yet. But this term here, the beta TV, are we going to view this as a birth of a new type of infection, right? So we're going to view E as a, or the beta TV, this rate, as a production of new infections. And what about the PI? Because this is a production of virus, right? So which of these two, or do we count both of these two, the beta TV and the PI, as new infections? And when we set up this next generation matrix, do we separate everything else into just transitions between states, right? Or are some of these also new infections, right? So how do we interpret our differential equation model in terms of reproduction of infectious individuals, right? And then what do we do with this beta 2 TV and how are we going to interpret it? And I was going to try and draw some pretty diagrams, but I did not. I don't have any that I could add into my PDF. And the way I have my computer set up, I don't want to try drawing. So if you take the view that both the binding of the virus to a cell to produce an infected cell and the production of virus here, if you treat both of those as infection, production of new infectious material, right? Then we're viewing a virus as this PI as a production of a virus, so a new virus appearing, makes sense, a new infection appearing. So it's a birth of an infection. But we're also viewing this beta TV as the birth of a new infectious cell, right? So if we view both of these as new, as fecundity, as birth, then we end up getting this reproduction number with this strange square root. And sorry, I didn't get a chance to write down the next generation matrix here, but the I guess it has an interpretation we can think about quite nicely. The numerator here inside the root has a beta, which is supposed to be this beta. This beta here is supposed to have a subscript 1. So the beta T is this rate of birth of new latently infected cells, right? The ratio of the two terms with the K is the ratio of this transition to infectiousness and the sum of the ways you leave E. So the K over the mu2 plus K is the time, expected time a cell spends in the eclipse stage, this latently infected stage. K, what's the other one? P is the rate that new virus is produced by an infected cell, right? And mu3 1 over mu3 is, we interpret that as a duration of infection, right? The time spent in the eye compartment, right? And so P over mu3 is the total amount of virus produced by a given infectious cell over its lifetime, right? Or average, I suppose, average over the cells. And then the 1 over mu4. So now we've basically looked at the appearance of the beta is the appearance of the cells as in this stage here as latently infected. The ratio of the kappa and the kappa plus mu2 is the amount that progressed to being fully infectious and productively infectious. The P over mu3 is the virus material produced by a infectious cell. And then this denominator that's left is just the time it takes before this virus is cleared, right? And so we're viewing this as two participants that are, the virus is using, is reproducing and giving offspring that are the exposed cells, and the infected cells are reproducing and giving new virus, right? So this reproduction number makes perfect sense, the same way the vector host one makes sense, right? Yeah, does that seem okay? Does anybody disagree? I'm not seeing any chat stuff. And this one is annoying because in the literature on the virology, the square root doesn't appear there, right? A lot of cases, because same reason that you end up with a square root randomly appearing and disappearing in vector host models, because the early papers just took the product of the two reproductions of the infected mosquitoes, basically, and the infected humans, multiplied them together to follow the cycle from human back to human, right? But if we look at this a little differently, okay, I'm not getting any complaints about this. Okay, so we can view this a little differently, and I like this because this is the ecological. This lets you put different interpretations on your equation and make up perfectly nice stories behind different reproduction numbers, right? So we can give an explanation to this reproduction number. It makes sense. It does. You can dive into the mathematics and prove that it's connected to the stability of the infection-free equilibrium, and away you go. You can use it as a threshold for whether or not the virus takes off in the host, right? But we could also view this as, I have to think for just a second. Yes, we can also view this in either of two ways. One is that it's the cells, the E and the I, are the mature states of the pathogen, right? So the pathogen, the pathogen is basically, the I is the adult stage of the pathogen, right? And so this PI is the production of propagules, right? In marine ecology, you look at all kinds of organisms that reproduce by producing propagules. Plants produce propagules, right? Seeds, various things that propagate somewhere else, right? So you can view these as either juveniles, the V, or propagules, right? They're the offspring that are now running around looking for, running around and needing to go through some process to mature from a propagule or a juvenile into an adult stage, right? And so the picture here is these propagules, right? The virus is running around looking for a cell. It then binds to and enters a cell, and then that's its reproductive stage, right? In the context of this TEIV model, right? And so if that's the case, this beta TV term is not a fecundity, it's a maturation, right? So the switch from free virus to virus bound inside the cell, you can make the analogy that this is a maturation of the virus. It's now going to grow into its reproductive form, right? Which is inside the host, hijacking the DNA and making replicates, right? If you do that, yes, baby viruses, yes. Except, yeah, well, actually, babies do run around and wreak havoc, don't they? Yes. Yeah. Oh, yeah. So this is a good question. So why is this last term in V prime negative? So both in in my analogy of these being juveniles or propagules, and with the fact that they're viruses, we're modeling viruses in a system. This is the binding of a virus to a cell. And so the V is our pool of free virus or intercellular virus. And E, think of E as a cell that has the virus inside it, right? And so essentially this virus has found a cell and found a home to mature in, right? Much like a larval green crab or lobster floats around, finds a place to settle, and then it can mature and survive in the adult form, right? Yep. So I got just a minute left here, right? If we do that, right? So that's why there's a connection between the beta TV here and this one here. And the reason the betas are different is you just might be measuring the V and the E in different scales, right? And so I think I'm going to go to this. If you do that, you get this reproduction number, right? And essentially one of the things I wanted to present and talk about was just I've been getting lots of questions of basically why is there this ambiguity, right? The same model can produce, if you make different decisions about how you view what the equation means, you get a different reproduction number, right? Surely one of those is correct and one of those is wrong. But my view on is they're both a result of a certain interpretation of the model. And so this one here, just quickly, the P is the reproduction, right? That's the birth of new juveniles or new free virus or little baby viruses, right? The beta 1T divided by this bit here essentially comes out as the fraction, well, it would be really a beta 2 times TV divided by this term here that would give you the fraction of virus that end up inside a host cell rather than like follow this route B2 as opposed to mu4. And yes, sorry, I have a bad habit of doing this. Capital T in this formula is the initial condition. So it's fixed at some level at the start, yeah. Except in a lot of the work I do, it is variable because we have different sizes of things, but never mind that. Yeah, so this capital T here and here, think of that as the initial condition, yes. Yeah, and then so the beta 1T over this piece here is connected to the fraction of viruses that find a host and a conversion between how we count viruses and how we count hosts, right? And then what's left? The kappas. The kappas is the same as before. The ratio of the two kappas is once the virus has found a home inside a host, what fraction of those mature to the adult stage here, right? Yeah, and so there's a perfectly reasonable description of both of these reproduction numbers. They're both thresholds for disease propagation, right? Or outbreaks of infection in the host, and they both make sense. So I think I was going to say a bunch more, but that's a good place to stop. I wasn't quite sure how much I'd get through. And that was the main message I wanted to get, was that there's nothing really odd about having different reproduction numbers for the same model. Oh yeah, so latently infectious and exposed in epidemiology. In epidemiology, think in terms of contact tracing, At least that's the way I think about it. If you are in the same location at the same time as a person who was found to be infectious, you are exposed, right? So in terms of the contact tracing, you're considered somebody who is exposed to the virus, right? That doesn't mean that you were infected. Or perhaps you did inhale some of the virus, but you didn't inhale a large enough quantity, or just because of variability in various things, the virus did not make it to your lungs and infect you, right? So you would be considered exposed, but never latently infected, right? It varies a lot from disease and pathogen to other diseases and pathogens, but essentially think of it as exposed just means you were physically exposed to somebody who is infectious. It doesn't mean you are actually infected, right? Does that make sense? Yeah. But in mathematical epidemiology, we usually, I don't know if we've changed, but we usually think of exposed as exposed to the infection and actually infected, right? So it's a different, historically the word exposed got used differently in epidemiology and in mathematical epidemiology. Different jargon. I used the word jargon in my first year calculus class, and they had no idea what it meant and thought it was hilarious. But yeah, it's just different jargon in different fields, right? So thanks. I should just quickly go to the end. Yeah, there. I just had some references to some papers, mostly not relevant because I didn't actually get to that section. I wasn't really thinking I would anyway. So thank you very much for your attention. And other questions?
Thank you very much. If there is any question, you can unmute, raise your hand and unmute your mic if you want to, or drop it on the chat. I think there's a question by Connor.
Hi, James.
Hi.
Thanks for the talk. Always instructive. The two values for R0, or the two expressions for R0 that you came up with, I didn't see them both at the same time. So I was unsure, is it just a square root, the difference in the expressions? Or was there more than?
Oh, it was.
Yeah. Okay. And then.
Oops. No, there's supposed to be, So there's actually a third one. There was another one I wanted to present that is actually, looks a fair bit different, right? Sorry, okay. I grabbed the wrong one when I prepared my talk. Yeah, okay. So actually on this one, it's just an explanation for that square root in some sense. Yeah.
Okay. So regardless of how different the expressions are, would it be valid to say that like you have the same set of equations that you're looking at either way, but in one case you're looking for the basic reproduction number of the virus, whereas in the other case you're looking for the basic reproduction number of a more generalized infection, which includes both types.
No, I think it's the same in the vector host models. You can, one is viewing sort of the vector and the host, or in this case, the virus and the infected cell on equal footing as agents, infectious agents, right? And the other is viewing them as different stages in the infection, right? And so in vector host models, when you take the perspective of looking at human infections, you can interpret your model as the infected host is just a different stage or somehow an offspring of the infected. So the infected vector is kind of a different stage of the infected host, right? And so to complete the cycle, you go all the way back to the infected host, right? So you view the infected host as the adult stage, and the infected vector is kind of a juvenile stage, right? And so you're thinking of a complete cycle, whereas if you view the host and the vector equally, you view it as sort of two connected cycles. Yeah. And yeah, and this also comes up in the, I had a nice one, that paper by Pauline. Yeah, this notion of the indirect transmission, that I think this is taken from paper of Pauline on cholera, right? I think. And yeah, you get two different ways of viewing things here that give you two different reproduction numbers. And it's a different views on what's going on with the free pathogen in the environment, right? So I was going to talk about following the pathogen and following the life cycle of the pathogen, but when you look too closely at the pathogen, so you were thinking, Connell, of taking the perspective of the reproduction of the pathogen, that pathogen for malaria, that life cycle is really complicated. It's got three completely different reproductive cycles in it, right? And you get, it's really hard to follow the pathogen through that. So I think the way to interpret it is you're following different interpretations of the life cycle of the pathogen, right? Or the life cycle of the infected individuals.
Okay, thanks.
Thank you.
I have these two.
Yes. Is there any other question? So I have a question for the in-host model. Is there any condition between beta 1 and beta 2? Like a comparison between them? One can be bigger than one.
I don't think so. And I think this does, so this actually happens in this model and in the model for cholera, or any model where you have an environmental stage of the pathogen. You end up with a freedom to pick the scale for V and E and I differently, right? You measure virus and you count the virus on a different scale with different units than you do E and I. So one easy way to think about it is that the two betas are there because they have different units, right? So there needs to be some conversion between units. But on the other side, in particular with the, right, and so if it's a conversion between units, that scaling is completely arbitrary, right? Depending on what you choose for your, how to measure the two things. Which, interestingly, I haven't got this sorted out to publish yet, but it's connected to what Pauline and Mark and Zhisheng, the third author, Zhisheng, did with the cholera pathogen. You get a type of matrix that doesn't quite fit this property that you're supposed to have with the invertibility of the matrix V and the sign pattern. But if you scale your variables, you can make that problem go away, right? And so it's the same thing happens here. But the other part, I think the answer to your question is that my understanding is you need to consume at least one virus, right? At least one virus has to be bound to a cell and enter the target cell, right? And so it's removed from, oh, sorry, I'm pointing with the wrong thing again. It's removed from the V line into the E, right? And so if you look at counting individual virus particles in individual cells, then it seems to me that beta 2 has to be at least as big as beta, right? You can't have a half a virus particle go in and infect a cell, I don't think. There's certainly lots of half virus particles running around, but I don't think any of them, in the sense you want, are infectious. Yeah, so in some sense that I think this, in that sense, this beta should be bigger than this beta, but there's a completely independent units, and so you can scale them however you want.
Okay, thank you. Is there any other question? I think it's time. I want to thank you very much for your great talk and taking time to give a talk at the Next Generation Seminar Series. Thank you very much.
Thank you for the invitation.
So, thank you very much for the audience for all the questions, and see you in the next seminar. Bye, everyone.